Okay, shall I wait a few minutes, Leah?
Let's go ahead and start with everyone. If you haven't had a chance to change your name or type your name into the chat so we can get an idea of who's here in attendance, that would be wonderful. And welcome to the Milo Baker chapter general meeting. Wendy Smith, who's our chapter president, will be starting us out this evening with a call for announcements.
Okay, well, I get started. I'm Wendy Smith, and I am the president of the Milo Baker chapter of the California Native Plant Society. I welcome everybody tonight. It's great to see all your faces, and I don't know how many of you are members, how many of you are from other parts of the state or the country or wherever, but whoever you are, welcome. And this is our second attempt at doing a virtual presentation. So I want to thank Chelsea for joining us and being willing to do a presentation on pretty short notice. So thank you, Chelsea. And also I want to thank Leah Giambastiani and Cody Ender, who have volunteered to fill in for Virginia, or with Virginia actually, while she does not have access to the internet right now, but hopefully we'll have her back. But Leah and Cody have been lining up presenters for the next few months, so hopefully we will have virtual presentations until we can all get together again at a regular meeting. Also want to say we had a very successful plant sale and 2 weeks ago, and want to thank all the members and volunteers who participated. They were our best customers, and we sold a lot of plants. We cleared out our shade house and gave some plants away to local nonprofits, one of which was the Laguna Foundation. And I heard that there was a big volunteer effort to get a lot of those plants put in the ground there on the property at the Laguna. So that's great news. So with that, I'll pass it on to anyone who has an announcement. Okay, Sean.
Hello, I just wanted to announce that we completed our fall sorry, I'm Zoom struck. Our fall
Scholarship.
Thank you. Our fall scholarship, and we received 7 applications, and 9 applications, and we were able to fund 7 of them. We feel very fortunate. We had a great list of applicants, both from Sonoma State and SRJC. So we'll have a little write-up in our newsletter to explain that. That's it. Thank you.
Thanks, Sean. I'd just like to give a sneak preview to the future speakers that we have coming. Next month, we've got Brooke Edwards from the Wildlands Conservancy, who will be talking about the Jenner Headlands Preserve. In January, we have Dr. Renata Toll from Davis and UC Davis. She'll be talking about diversity in conservation. In February, we have Wendy Herneman, who is one of our scholarship recipients, and she'll be talking about her research work in oaks. In March, we have Amelia Ryan from Pinnacles, who will be talking about her work at Pinnacles. In April, that was March. In April, we have Lorraine Parsons talking about microbial communities in dune restoration. In May, we have Trish Tartarian, ecology of the California tiger salamander. And then in June, we have Shane Hanafy, botanizing Nevada and Placer counties. So lots of great speakers to come. And the one we have come today too.
Okay, anyone else have any announcements to make? Okay. All right. Well, take it away, Leah. Hopefully you're ready to introduce our speaker.
Yes. So I have the pleasure of actually getting to work with Dr. Chelsea Carey. She's a Working Lands Research Director and Principal Soil Ecologist at Point Blue Conservation Science. And I've had the pleasure of working with her through a project where we're doing soil inoculation, taking microbial communities in same types of species, the same species, and moving it to an area that doesn't have any of those species in order to bring that microbial community and bring those benefits. So I just want to say it's an exciting project. It's an experiment that we're working on, and we'll see how it works out. But I love having the soil perspective in the restoration work we do. Chelsea, Carey, forgive me, I also have kids coming in, so if there's a little noise in the background, that's why. Let's see, Chelsea Carey, as our Working Lands and Research Director, has developed and led priority research projects and partnerships that help inform rangeland management across the state. And she generally focuses on characterizing soil properties that are relevant for rangeland soil health and climate change mitigation, and determining how the management influences these properties across space and time. Chelsea, I'm actually going to let you help fill in the rest of the introduction. Awesome. Thanks so much for joining us tonight, and looking forward to hearing your presentation.
Thank you, Leah. Thank you so much for inviting me to spend the evening with you all. I'm really excited to be here. So I will screen share here, and then I'll continue the introduction, and we can get on our way. So let me share my screen, and then let me present. Okay, I'm assuming you can see my screen. Is that correct? Yes, okay, I see some thumbs up. So yeah, so super excited to be here. As Leah said, I am the Working Lands Research Director and Principal Soil Ecologist at Point Blue. I've been with Point Blue for just about 3 years now, and prior to joining the organization, I got my PhD from the University of California Merced, where I studied nitrogen cycling and microbial community dynamics in California grasslands, and I'll talk a little bit about that work today. I then spent some time at UC Riverside, where I focused mostly on understanding dispersal mechanisms for microorganisms regionally and globally in the atmosphere. But I also did some work on Brachypodium distachyon and trying to understand how it is influenced by microbial communities in its native range in Spain, but then also in its introduced range here in California. So I won't touch on that work today, but I have spent the past 10 plus years thinking about native plant conservation. I wouldn't, and when I was reflecting on this presentation, I didn't think of it as such, but when I look back at what I've been doing, a lot of it does have to do with sort of native plant conservation and restoration and that below ground connection. And so I will start today by just talking generally about plant-soil interactions and what they mean for native plants and how we study these interactions to better understand the importance and implications. And then I'll sort of highlight 3 case studies that I've worked on that I'm currently working on. And that includes work on the giant Sequoia. It includes some work from my dissertation when I was in my PhD looking at grasses and exotic grasses and nutrient availability. And then I will highlight and finish up by talking a little bit more about that project that Leah just described that we're currently working on. I won't share any results from that, but I'll share a lot of pictures and then we'll wrap up. So, I need to have you guys, I minimized you, but then I'm just talking to the ether, so I have to have faces up still a little bit. So soils are highly variable across space and time. And, you know, and plants are sensitive to that variability. And so on the left here, it's just a schematic of sort of how you have layers of spatial variability in soil properties. So at the landscape scale, you have variation in soil texture, soil carbon. You also then sort of layer onto that plot level or field level variability from burrowing animals. And then at the micro scale, you have the interactions between roots and the soil surface and microorganisms and differences in aggregation. And you sort of layer on top of that, and you can quickly begin to see how much spatial variability there is in soil properties. You also have variation across time in a lot of these properties as well. Everything from variation in the organisms who are within the soil. So this is just some illustrations from a guide that I put together with a colleague. So just as an example, right, you have spatial variability in nematodes, which is pictured in the top left on the right-hand side. You have spatial variability in things like nutrients, which are really important to plants. That also is going to change over time as well. And then on the bottom, you can layer on top of that variability in things like soil texture and water, or oxygen availability and soil structure. And all of these properties are known to be important to plants, and plants can prefer certain conditions over others. And so that's, that leads then to variability in plant communities across the landscape, and is why we have, like indicator species. So you can have plants that indicate that you are on very wet landscapes, right? You can have plants that are early or late successional species. And so plants respond to soil, but once they're established, plants also influence the soil, right? So they have particular traits like water or nutrient uptake, litter quality, and things like rooting depth. And so there's a lot of different traits we can think about and how they might influence the soil or the ecosystem once established. And when we talk about litter quality, we are talking about things like the carbon to nitrogen ratio of the litter, or the amount of lignin in the litter. So how easily does that litter, for instance, decompose? And so here's just one example, right? So this is on the left-hand side, pictures of yellow star thistle root growth. And we know that yellow star thistle consumes a considerable amount of water, and it does so with this taproot that goes really deep into the soil. And so on the right-hand side is some work from Stephanie Enlow showing that this taproot from yellow star thistle can extract a considerable amount of water, such that you see sort of differences in water content through the soil profile when you compare it to other annual grasses in California, and there's this one particular perennial grass they were looking at. And so on the x-axis here is soil depth, the y-axis is water content, and these closed circles is water content associated with yellow star thistle. And you see, especially as you go deeper, it's lower, and the water content's lower because yellow star thistle has that deep taproot and it's extracting the water, so water content's lower. So this is just one easy example of how plants differ in how they have in their traits, and how that influences a soil property like water content. Another example is this litter quality. And so again, when we think about litter quality, you think about things like the amount of nitrogen to carbon, and the amount of lignin, for instance. Things like silica content and stuff can also play a role. And the quality is going to affect how quickly this litter decomposes. It can affect a whole lot of other things, but one easy example is the rate of nitrogen cycling through nitrogen mineralization and decomposition. And so on the left-hand side, right, you might have an oak tree or an oak species that has litter that decomposes is much higher in quality when you compare it to something like a Doug fir, some kind of conifer. And so that's going to have implications for nutrient cycling within the soil profile through differences in those litter quality traits. So again, just another example of how plants can influence that soil environment once they establish. So you have plants that are responding to soil. You also have then these plants influencing the soil. And so when you have this response and this influence, what you can get is this feedback, this plant soil feedback. And plant soil feedbacks can shift the competitive balance between species that are present within a community and ultimately alter community dynamics, so plant composition, for instance, or diversity. So this is just a figure illustrating ways in which native and exotic species can sort of change the soil and influence their own growth or the growth of a neighboring individual. And so in a lot of cases, native plants actually promote, they can promote sort of these antagonistic or pathogenic soil organisms, and that helps to keep them in check, right? So it like keeps, in a lot of cases, them from, you know, becoming weedy. That's why they can coexist with other organisms. It's one reason. Exotic species or that have been brought over from, you know, maybe Eurasia, they one mechanism for them to become invasive and sort of spread in an uncontrolled manner is through this enemy release hypothesis. And so if they're in this introduced range, but their enemies, their pathogens, which they have co-evolved with, have not been transferred with them, they essentially we find them in a situation where they can have these beneficial relationships with organisms like mycorrhizal fungi, but they don't have that pathogenic pressure, and that can contribute in some cases to their success. Now, this can directly feed back to influence sort of those other plants within their same species, but they can create changes within the soil then that influence, like I said, co-occurring species as well. And so this can, I think, is one thing that's really important to recognize when we're trying to understand sort of the success of native species and the success of projects where we're trying to restore native plants is how are these organisms and these qualities below ground, whether it's nutrient dynamics, or microorganisms, how are they being affected by the plant species that are there, and how is that feeding back then to the success of the desirable plant species. What's more is that sort of when a plant establishes a population at a given site, and in this case, I've just generated a schematic of sort of this potting experiment, but plants can essentially condition the soil, right? So they can alter it in ways that are perhaps beneficial to itself. In some cases, maybe not, but what happens is when you change those, and if it's different than what a desirable or a native plant might want, then you can have these legacies. Those changes might even remain within the soil even after you remove that undesirable plant. So this undesirable plant, let's say it's an invasive or something, it can establish, it can alter the soil. So in a lot of cases, invasive plants, like grassland plants, they increase nitrogen availability in California. They can shift the microbial community. Even if you remove them, those changes can remain. And so if you try to seed in native plants in that conditioned soil that sort of is imprinted, has these legacies imprinted, that can challenge the success of that reseeding, right? So it can challenge the success of that native plant is if that native plant likes conditions, soil conditions that are different than what is there. So often native plants we think of as preferring lower nitrogen availability, and of course this is a generalization, but lower nitrogen availability, they might harbor different microbial communities within the soil that they prefer. And so we have shifted those communities, they no longer exist, and that's going to make it challenging for those native plants to be successful. And ultimately what you might see is if you are in not a potted experiment like this, but out, in a pasture, for instance, is that then that undesirable plant, because the conditions are favorable to it, will be able to reestablish because it has that sort of competitive edge. The conditions are favorable for it and not for your desirable, perhaps native plant. So that's sort of like high level, some of the thinking around why plant soil interactions might be important for things like native plant conservation and restoration. There's a lot of ways that we can study plant soil interactions. And I just wanted to like highlight a couple of them. I think it sets the stage for the 3 projects I'm going to talk about. So the first way that we can study plant soil interactions is observationally. So you can go out onto the landscape and look at native grasses and non-native grasses. And you can ask, how do they differ in soil properties, right? And in this case, I've just provided an example with microbial communities. And so these little circles and triangles and such are different microbial species. And so in this hypothetical case, the perennial grasses have greater microbial diversity than beneath the annual grasses. And so we can just go out observationally and see whether or not that this is the case on the landscape, right? It allows us to sort of get at scale, which is one benefit of this type of observational experiment, and it allows us to generate hypotheses. But what you can't do with something like that is get at causation. So you can't know definitively whether or not the perennial grasses promote more microbial diversity or they respond to it, right? So maybe they preferentially establish on sites that already have more diverse microbial communities or other soil conditions. Or maybe they don't, and actually over time they're promoting that. Again, this is the hard part because plants respond but also influence the soil. And so one way to get at that is through experimental approaches that allow us to ask sort of whether perennials promote a more diverse microbial community through like common garden experiments, right? So you can take plots that didn't have perennial grasses, didn't have these annual grasses, you can do them in containers or in the field, and you seed, right? And so you establish these communities and you look at how the soil properties that you care about change over time. And so that allows you to understand whether or not any differences you're seeing observationally are actually being caused by those plants. And then one of the final ways that we can look at sort of plant soil interactions is by another experimental sort of approach, so in this example, we know that perennial grasses differ in microbial communities from annual grasses from that first observational experiment. And the second one, we're showing in this example that perennials are actually conditioning and creating that more diverse microbial community. And then the third sort of thing that one might want to do is ask, well, does that matter, right? So if microbial communities do differ below perennial native grasses and annual grasses, like, does that matter for plant success or any sort of other outcome you might care about? And so there you can, grow, for instance, perennial grasses, the same perennial grasses, again, in sort of a common garden, but you might want to inoculate with a more diverse microbial community and a less diverse microbial community, or change those soil parameters that you think might matter, and you can see how your plants do in response, right? And so in this case, when you grow the perennial grasses on a more diverse microbial community, if you can tell, the plants are bigger. And so then in the soil that had sort of this less diverse microbial community in this example. And so you're able to get at from sort of observational to mechanisms, and then to sort of inference about the ecological implications of those differences. And so the three examples I'm going to walk through, they are all different systems, but they sort of track nicely onto these different examples. And so the first observational approach, I'll sort of walk through something similar to that we did with giant sequoia. I will then talk sort of about this like conditioning, or these very controlled experiments that we're able to get at a little bit more causal relationships with native versus invasive grasses in California. And then the final one is this experiment that I'm doing with Leah and others at Point Blue, which is inoculating with microbial consortia to essentially see whether or not that helps the same plant species establish in restoration projects and survive things like climate stressors like drought into the future. Okay, so the first example, is, like I said, we're looking at soil microbial communities associated with giant sequoia. And this was a fun project. It's actually one of the first ones to ever look at just, so there's been a little bit of work done on giant sequoia and their mycorrhizal partners. So giant sequoia associate with a type of mycorrhizae called arbuscular mycorrhizae. And this contrasts with a lot of other trees in the forests around them who associate with ectomycorrhizal fungi. But beyond that, we don't know much, or we didn't know much about the microbial communities in the soil associated with giant sequoia, nor did we know much really about sort of the soil properties associated with giant sequoia as well. There was a couple of studies decades ago. And so there was real opportunity, we thought, to just do some observational experiments where we went out to 2 groves in Yosemite. So we went to Mariposa Grove and we went to Merced Grove. And we sampled beneath giant sequoia trees in each of those groves. And we also sampled beneath co-occurring sugar pine trees. So we had a sort of a comparison to compare the giant sequoia microbial communities and soil properties too. Well, I think before I go on, I think what I will say too, you know, the microbial communities are super, like, soil microorganisms are really, really important. Soil microorganisms help, you know, plants to you know, they create sort of mutualisms with plants that help them to grow and you know, withstand stressors. They help to make nutrients available to plants. There are in a handful of soil, and I'm sure most of us have heard this, but in a handful of soil, there's, you know, more microorganisms than there are people on the entire planet. And we know they're super important, and we still have a lot to learn about what most of them do. And so I think that was another motivator for this, is that, you know, you have this iconic giant sequoia that's long lived, and we like the microbial communities associated with them must be important to some extent, but because they're important, you know, in most cases. And so I think that there is just an opportunity here to learn more about those really critical microscopic players. And so, but before I get into the microbial community side of things, we did also assess soils associated with giant sequoia and that co-occurring sugar pine across those 2 groves. And this is just sort of a panel of different soil properties. And so everything, so the top, it has aluminum concentrations. The next one is nitrogen availability, that AMN. You have BD is bulk density, so how dense the soils are. Gravimetric, the GWC is gravimetric water content, so that's soil moisture. You have ammonium, so nitrogen availability, pH, then sulfur. Then you have cation availability. And then on that bottom, you have carbon, total carbon and total nitrogen. And the giant Sequoia samples are that left-hand box, and then the sugar pine are on the right in all of those panels. And in most cases, what you can see is that actually giant Sequoia has higher amounts or concentrations or values of those properties, in which in many cases is sort of, you can think of it as like kind of like being cushier. Like that's like they create some like really nice conditions for themselves. So you know, these are acidic soils, they're making them a little bit less acidic. They you know, are increasing, they have like higher nutrient availability and cation availability. Their soils are not as dense and they have more soil moisture. So compared to this sugar pine that's co-occurring with it across these groves, you do see this effect at the in these soil properties, and I would say it's leaning towards these sort of cushier conditions. We looked at, so in addition to these soil properties, we looked at bacteria and fungi. And we did this using DNA amplicon sequencing. So we're able to look at essentially who is there in the soil community. We can ask things like about community composition. And then we also can ask things about like diversity, like richness. And so I will walk you through these. So on the left-hand side are figures, and I probably should have simplified this, I apologize. But they all essentially say a little bit similar story. So if we just focus on one panel, like the upper left-hand panel, that's for bacteria and archaea, and this is an ordination plot. So for those who aren't familiar, so this is taking all of the organisms that are in a given sample, of which there are thousands. So in a given sample, soil sample we take from beneath the giant Sequoia, there are thousands of bacteria or thousands of fungi. And how do you make sense of thousands of species? Well, what you can do is you can kind of take all that's in there and you can scrunch it down into this little sort of like 2D space. And for a soil sample then that has a community that is similar to another community, those spaces, those points are going to be close to each other. So if you look at the like two blue points and they're close to each other, that means that that's two soil samples and their communities of bacteria are similar to each other. Points on that space that are far apart from each other are more dissimilar in their community composition for bacteria. And these are color coded by tree species, with the sugar pine as the red and the giant Sequoia as the blue. And then it's also within the two different groves. And what we would expect if communities were the same across tree species is that the red ovals and the blue ovals would overlap. But they don't. They're actually sort of separated. They're kind of pulled apart. They're not overlapping. And what that says is that the bacterial and fungal communities beneath giant Sequoia and beneath sugar pine differ when you take into account those thousands of species that are there. It doesn't tell us who is driving that difference, right? So what species within bacteria are differing, but it just says when you take you know, sort of consider all of it as a lump sum, that they differ. We can also ask then about richness. So this is just the number of species. So it's often what I think people think of when they say diversity, is like the number of species. And that's what OTU richness means. So on the top, the giant Sequoia is the blue and the sugar pine is the red. And you can see that across both groves, giant Sequoia harbored more bacterial species, so they were more diverse than the sugar pine. There was not a similar trend with fungal richness, which was interesting, but we saw it consistently with bacteria. So giant Sequoia have greater bacterial richness or diversities than sugar pine. And then I think this is the last figure I'll show from this. So I mentioned at the beginning of this portion that giant sequoia are known to associate with arbuscular mycorrhizal fungi. So there are two types of mycorrhizal fungi. There are arbuscular mycorrhizal fungi. Well, there's more than two, but there's two common kinds. There's arbuscular mycorrhizal fungi, and then there's ectomycorrhizal fungi. And they differ in how they form associations with plant roots. And that's what this little figure is showing on that little diagram is showing on the left. I realize now that it's actually maybe a little bit too small. But arbuscular mycorrhizal fungi, they penetrate root cells and they exchange nutrients for carbon. So these mycorrhizal fungi, right, they're giving nutrients and water to the plant. They essentially act as extensions of the plant root system, they're smaller than plant roots, so they can get into little crevices in the soil that plant roots can't and access water and nutrients. And they give that to the plant, and the plant in return gives the fungi the carbon that they need to grow and survive. And so it's what we call as a mutualism. And the AMF, the arbuscular mycorrhizal fungi, do it by penetrating the plant root cells. The ectomycorrhizal fungi do it in a little bit different way. They don't penetrate the plant root cells. They sort of form these other exchange sites. And most plants, most grasses, and a lot of trees actually in different ecosystems associated with arbuscular mycorrhizal fungi. And then there are some other trees that associate with ectomycorrhizal fungi. In the Sierras, a lot of the trees associate with ecto, but giant sequoia associates with arbuscular mycorrhizae. And so then there's so we would expect that you would see signatures within the sort of sequencing that we did, the DNA sequencing, such that the sugar pine had more ectomycorrhizal fungi in the DNA sequences, and giant sequoia had more arbuscular mycorrhizal in their community. And so that's what, and we do see that. So it's kind of like a nice sanity check. And it's interesting to see, because we weren't targeting plant roots, it's interesting to see that it's sort of extending into what we call the bulk soil. So just sort of the soil that is, can pick up with your hands in many cases. And so on the left-hand side, it's we're looking at the relative abundance. So how abundant are the ectomycorrhizal fungi within these samples? And you do see that they are more abundant beneath sugar pine than giant sequoia, and that's what we would expect. And then the arbuscular mycorrhizal fungi are more diverse beneath the giant sequoia. And again, this is what we would expect, but it's always nice to see your expectations confirmed. So I think the takeaways from this is that there's considerable differences between these two plant species, and there's possibly, tied to giant sequoia's longevity and immense size. They have much greater diversity of bacteria. They also have, and so this diversity of bacteria and fungi may aid in the long-term success of giant sequoia. There's some of these differences, they can actually act as like seeds for endophytes, so organisms living inside these plants that help them grow and survive stressful conditions. And so I think that there's a lot there to dive into, but the fact that they do harbor sort of this immense diversity is really intriguing. And it also says that if we lose giant sequoia, so giant sequoia is threatened by things like fire suppression, it's threatened by droughts, that, this would likely have an effect on below ground biodiversity because they are harboring more, or sort of a more diverse community than some of their co-occurring species. Okay, I'm gonna plow on. I wish if we were in person, I would make this more interactive. Hopefully folks are still with me. So the next project I want to touch on, and there's going to be many less figures in this one, is some of my dissertation work. So looking at comparing invasive grassland plants to naturalized plants, so exotic plants in California's grasslands that have been here for a really long time and are sort of widespread, to native plants, some plants that we try to sort of target for restoration in a lot of places. And I focused on nitrogen dynamics to a large degree. And I did that because nitrogen is super important. It's a plant, it limits plant growth in many terrestrial systems. And so changes in nitrogen can have large effects on things like plant community dynamics. I will say too that, you know, in California, there has been a decent amount of work looking at the difference between annual grasses and perennial grasses. And often what we find in a lot of cases is that annuals have faster sort of increased nitrogen availability and have faster rates of cycling compared to perennial grasses. And this is a phenomenon that we see across the board with things like invasive plants, right? They tend to sort of be what we call nitrophilous, so like nitrogen loving, and they have traits that allow them to essentially speed up nitrogen cycling and increase availability, which again can shift the competitive advantage in their favor. And so I think one of the motivating questions for me with this project was, well, do we see that with some of these newer wave, what I call newer wave invasive? So there's been a decent amount of work looking at these naturalized exotic, what I call naturalized exotic species. So your wild oats, your bromes, and comparing them with some of your native species. But then you have, you know, Barb, goatgrass, and medusahead that are spreading throughout the state right now. So these naturalized species, you know, they are widespread. We typically don't manage to try to remove them, right? They're part of the landscape. But these newer wave invasives, they, you know, are often like a high priority in terms of like Calypsy rating. They are managed for or against in a lot of cases still. And so I think that there's a lot of motivation to understand sort of the driving mechanisms for the invasion, and then also the consequences, the ecosystem consequences. Barb, goatgrass, and medusahead are interesting because they both create sort of like thatchy layers. If I don't know how familiar folks are with them, but they create this like thatch. They don't decompose really well, and it's actually because they have pretty poor litter quality. And so one has really high silica content, the other has like really like a lot of lignin, high carbon to nitrogen ratios, and that can slow its decomposition. And that thatch layer is thought to actually, they can germinate underneath it, but you know, native plants and others have a harder time. And so that may be one primary mechanism for their, or one of the mechanisms for their success. But that kind of dynamic and that kind of change again in litter decomposition should have subsequent effects on things like nitrogen cycling or nutrient cycling. And of course, then the question is, well, does that help to facilitate their invasion? And so I set up this, so I worked in Davis before I go through that. So for my dissertation, this project was not set up by me. It was a very beautiful project. This is an aerial image of all of the plots. So this is where it was a controlled experiment. And there was a whole lot of treatments. And it was led by some researchers at Davis, so Valerie Ebener, and then Carolyn Malmstrom and Kevin Rice. And they were, you know, interested in understanding how changes in plant communities influence, you know, ecosystem dynamics, but then also you can see some rain shelters on there. So they were interested in looking at sort of the interaction with changes in precipitation, and there was a number of other treatments on top of there. But so I had these, you know, at my disposal. What I had was this really beautiful experiment where there were these 3 different plant communities established for something, I think it was about 6 years when I started my experiment. so a relatively long time that then I could ask questions about nutrient cycling in those different experimental plots. And I'm just going to show one figure. So this is, so I was able to look at nitrogen cycling across time. And I did this for a number of years. And this is from, I can't remember off the top of my head which year this was, but it was going from it was a water year, right? So starting in November, going through August, looking at, this was nitrate availability. So a nitrate being one of the plant available forms of nitrogen in the soil, and how its availability and sort of cycling is affected by across these different treatments, and fairly consistently, especially during sort of the rainy part of the year, there were strong differences in availability such that you saw a decrease in availability in invaded communities. So these communities of Barb, goatgrass, and medusahead compared to natives. The effect, sort of the difference between invaded communities and naturalized communities were not as strong, but it is interesting to see sort of across the board that the naturalized communities, so again, these bromes, the oats, they're falling in between in a lot of ways, these invaded and native communities, which is really interesting to me. And so here, this is sort of what we would expect. So they're reducing decomposition. There is this sort of ultimate effect on nitrogen availability within these invaded communities when you compare them to native grasses. And when we looked at a whole other suite of nitrogen cycling metrics, we saw a very similar dynamics. So we looked at potential rates of nitrogen cycling in the laboratory, for instance, and again, sort of, it was all confirming the same picture. And so then the question is, right, so so we're able to essentially demonstrate that not only sort of would we expect to see differences observationally in the field, but that these nitrogen cycling changes are happening as a result of sort of who is there in the plant community. And I would argue that there's possible consequences for things like native plant restoration. So when invasive plants increase nitrogen cycling, often people will have to try to restore the soil before they can restore native plant communities. They will put down sugar, or they will put down wood chips. They will, essentially, that carbon source can suck up that extra nitrogen and remediate the soil before then you try to reestablish your native plants. This is the opposite direction, but could require similar types of intervention if somebody is wanting to sort of remediate these invaded sites by reestablishing native plants. I thought it was also interesting that very few differences were detected between invaded and naturalized communities. And so this suggests to me that the below-ground effects of Barb, goatgrass, and medusahead invasion, like across the landscape, because most of our landscape is covered by these naturalized species, not the native species, then the invasion is perhaps having less of a dramatic or pronounced effect on nitrogen cycling than we previously thought. But it's important, again, if we want to remediate and ultimately get back to those native plants, that we do need to consider these nitrogen dynamics. Okay, and so I will finish by describing a project, and I will show no graphs in this one, only pictures. Okay, so this is a fun one. This is one that we're working on right now. And we're calling it Inoculant Supported Restoration of California's Rangeland Riparian Corridors. If we think again about these mycorrhizal fungi, because they allow plants to access additional nutrients and water, they can also do things like help to minimize pathogen loads and pests, on plant roots. They have a lot of demonstrated benefits for plant growth and survival. And so this is just one picture I found off the internet of a plant that had a mycorrhizal inoculum and then no mycorrhizae. And you can see this time and again, that sort of plant biomass is often going to be larger in those that are inoculated than those that aren't. There's sort of emerging and a considerable literature showing that, you know, mycorrhizal partners also confer things like, you know, tolerance to stress. So whether that's a drought or salt or or heat, that these partners are really important. I already talked about the difference between arbuscular and ectomycorrhizae, but I wanted to highlight it again because here, in these restoration projects, so we're planting so it's in partnership with the STRAW program that Leah is part of, so Students and Teachers Restoring a Watershed. And in these restorations that they put out on the landscape, the plants, many of them associate with arbuscular mycorrhizae, but some of them prefer to associate with ectomycorrhizae. And it is those that prefer to associate with ectomycorrhizae that we are targeting for this experiment. So because most plants associate with arbuscular mycorrhizae, grasses, for instance, you can imagine that if you have a site like this, that is all grasses, it hasn't had trees for, you know, I don't know how long, but, you know, let's assume it's a considerable amount of time, then, and the trees are not close, so there are some in this picture, what you would expect then is that because you don't have plant hosts for these ectomycorrhizal species, that they might be, there might not be many viable ectomycorrhizal spores, for instance, in hanging out in the soil, ready to form those mutualisms with plants when we plant them, if that makes sense. And so, but when we put the arbuscular mycorrhizal plants in, they can take advantage of all of those arbuscular mycorrhizal fungi that are associating right now in this picture with those grasses. But if we're putting plants that need different types of mycorrhizae that don't associate with those grasses, then one could hypothesize that introducing them with those particular plants would help those plants do better in the near term and the long term. You can sort of go a step further and make some hypotheses about where you get the, how effective those mycorrhizae might be depending on where you get them. And so, so what we were motivated to do was to get two different sources of these ectomycorrhizal inoculum. And we got these we took them from the field ourselves. So essentially we collected soil from beneath reference sites, like really like established riparian areas that have the tree species that we're interested in. You take soil from beneath them, theoretically that should have the ectomycorrhizae that we're interested in introducing. And we did that. We took sort of two different types of inoculum. So we took it from nearby reference sites that have similar climate to where we were restoring, and then we actually found some sites that were hotter and drier in the region. And there is some evidence that, you know, so mycorrhizae can have adaptations just like plants and other organisms can. And so mycorrhizae who have sort of evolved in hotter and drier conditions, or been hanging out in hotter and drier conditions, they might have some traits that allow them to do better in those hotter and drier conditions that are expected to occur where we are doing restoration, right? So we're gonna have increased drought. We're gonna have increases in temperature. We know that from predictions, right? And so can we introduce mycorrhizal fungi that are already adapted to those conditions that are projected for these sites? And if we do that, do they help the plants do better, or does it not really matter? Or maybe they do worse because those conditions aren't here yet, right? But I think it's an open question. And so what we did was we had these two different inoculum sources. And this is just sort of a schematic of how we did it. So we looked, so these were the four species, plant species at the top that we targeted. So we targeted coast live oak, valley oak, Doug fir, and white alder. Sorry, not white alder. Willows. We targeted willows, not white alder. And we went to those reference sites, so either the hot dry or the nearby site, and we kept them separate, the collections, but we sort of collected soil, bulked them, and then we, when we did our plantings at these sites, we introduced sort of those soils in the communities that they harbored with the acorns or with the Doug firs or with the willow sprigs. And so what that looks like here, and so we did also test to make sure that we weren't introducing some bad pathogens with it. So we first did pair testing, pair baiting on the soil samples to make sure that they were free of Phytophthora. And then we created an inoculum, and the inoculum was really sort of some of the leachate from the pair baiting, and it was the whole soil. So it was a soil that we collected from beneath these trees in the reference ecosystems. And with students and with straw staff, we had a number of really fun planting days, and it was such an awesome outreach, I think, opportunity to be able to, and a teachable moment for students to be able to learn about these microorganisms in addition to getting the experience that they get with straw anyways. So we did a bunch of plantings and where we introduced the inoculum as well, and we have treatments, right? So we have a no control, and then we have these two different inoculum treatments, and now we're in the monitoring phase. So on the right-hand side is one of the straw staff, Isaiah, and he is monitoring a willow. And so we do have data, and none of it is quite together enough yet to be able to present any of it today. But maybe I can come back and share the results with y'all at some future date. So here, the takeaways, I just wanted to add a takeaway because I like to be consistent, and I had them on my other side. So, mycorrhizal fungi are super important. They're going to become increasingly important, I think, with more frequent and intense droughts and other climate stressors. In some cases, mycorrhizal inoculation may help native plants to survive restoration. It could decrease costs associated with maintenance, right? So that they help to increase water access for these plants, then perhaps we have to do less watering. And then the source of inoculum could influence the magnitude of the benefits again, but I would say stay tuned, and I'll look forward to sharing what we find, at some point in the future. And so I'll just end by first saying thank you, asking if there's any questions, and giving a little plug if some of the illustrations that I highlighted throughout this presentation are actually from one of our colleagues at Point Blue, Mel Preston. She's an amazing scientific illustrator, and I've had the pleasure of producing one outreach material with her on the right-hand side, so Life Below Ground on the Range. This is all about soil communities in California in particular, focusing on rangeland systems. And then she also produces beautiful pasture plants guide for specifically for San Mateo County. And we have copies of those. I'm happy to share both online and hard copies. So.
Thank you so much, Chelsea. Really fascinating information that you put together. So far, I've just seen one question in the chat from Wendy Smith. Wendy, do you want to share your question?
Wendy is muted.
All right, I'm going to read her question. She was asking, I believe, about the arbuscular and ectomycorrhizal communities, and she said, do the two species have different root structures? That was it.
Yeah, I think I would need to know which two species is this.
Oh,
so
sorry.
No, I was referring to the part of your presentation where you talked about the sequoia versus the sugar pine, and whether the differing root structures between them would have an influence as well.
I think that's a really good question. And I honestly, I don't know much about their rooting structures, and so I'll have to punt on that question. And I honestly it's interesting because the studies that have been done on giant sequoias like below ground are, like I said, very minimal. So there's some studies from a couple of decades ago, and they were looking at changes in soil properties as you move away from the trunk of the tree. So you can look at sort of in space how soil properties move as you go sort of, yeah, from the trunk of the tree outside of the crown. But everything that I have seen doesn't go very deep. And so somebody who probably knows more about just tree structure in general than I do might know, thinking about the different sort of families or, yeah, but off the top of my head, I don't know. And it's a good question.
I think we have some other questions that have popped up in the chat. Let's see, Guido, would you like to unmute yourself and share your question?
Yeah, thanks, Leah. Hi, Chelsea. Thanks so much for the presentation.
Thanks.
I was just curious about the AMF and EMF and, well, first, how does compost really change, soil composition for natives, as there's a lot of proponents, saying about spreading compost. And the other one is it possible to inoculate compost with AMF and EMF and spread it? And if so, have you know any studies or anybody that's experimented? Or is it possible to just use compost tea during the rains?
Yeah, it's a really good question. I mean, my understanding of compost and compost tea is that like a lot of it, like what you're selecting for are going to be like saprotrophs. Like, so you're going to be like selecting for like, not necessarily mycorrhizal mutualists, but, free living sort of fungi that help to decompose. I don't know of anything sort of in terms of the composting process and selecting for or against AMF and EMF. So even like, David Johnson's bio reactor is Beam. I haven't seen much discussion around selection necessarily for mycorrhizal fungi. What I hear is a lot about the sort of the diversity in general of fungi in those composts. And so I think, I think conceivably you would, especially if they're spores, but it's a good question. And I haven't seen much on it. I think, in a lot of cases, like if you're trying to inoculate with AMF and EMF, I might not even compost or compost tea it. I might try to do some sort of straight inoculum that doesn't go through a composting process. Like we did.
I know compost typically gets, to a pretty high temperature for a period of time in order to,
Yeah, this would be adding it after the composting process. So like almost like during an aging process before it's spread on pasture.
I mean, you could. I don't know. I haven't heard of that. But I don't see why not. I mean, I think that there's a lot of different ways that you can create your own sort of inoculum, and you can do different extraction methods that vary in sort of rigor or like intensity. And I think that I don't see why you couldn't try to add in some of like AMF or EMF spores from, for instance, like reference sites at your area. Now EMF, in terms of like commercial inoculum, EMF is hard to come by. AMF is the one that's, I mean, when you get mycorrhizal inoculum from commercially, it's by and large going to be AMF. So if you were trying to get EMF, you might need to go and take it from like a reference site.
Sorry, just one more thing I want to add, because it was something that was really important with the work we did with Chelsea when moving soils. Just be really careful about possibly transporting pathogens from one spot to another. So we wouldn't want to spread, our very famous sudden oak death, but there's actually more than 30 species. Might even be in the hundreds now, but of Phytophthora. So those are pretty deadly for plants. So we definitely don't want to be moving those around without doing some testing.
Yeah, totally. And and I don't know, and the pair baiting, method has its pluses and minuses, and but is relatively, I think, accessible as an approach. So for what it's worth. But yeah, I would second what Leah just said.
Thanks. Okay, next question. We have, how long has the riparian restoration study been going on, and how long will monitoring continue?
Yeah, so it started, it was awarded from the Wildlife Conservation Society, actually. They have a climate adaptation and mitigation fund, and we got it funded last fall, so fall of 2019, and we hit the ground running. So it's a 3-year grant, and we needed to get implementation in immediately. So all the implementation happened this winter. And this is our first monitoring right now, and it will be going through through 2021 to sort of the winter of to like January or February of 2022, and we'll monitor through that. And the awesome thing though about it, this, sort of leveraging the straw program for some of this work is that, they, like Leah and others, have such amazing relationships with these producers. And so, the hope would be is if we're seeing some cool results, or if we want to go back in the future, those can be conversations we have to sort of do some longer term monitoring.
Thanks, Chelsea. I should have let Wendy ask her own question for that one, but Alina, would you like to ask the next one? If you're unable.
Sure, I will ask that question. In walking around post-fire, and especially in last year walking around in a post-fire and thereafter post-logging area, I noticed that the resprouting vegetation that came up, especially under the areas that had been burn piles, were remarkably stunted. And based on what you said earlier about mycorrhizae being vitally important to plant vigor, I wondered if that was potentially the result of that effect, or if there might be something else going on, like the nutrient cycling that came from the burn pile itself might have an effect.
Yeah,
It's
a good question. I mean, it's hard to say. So it certainly could be something microbial. It's depending on like sort of the severity of the fire. So the more intense it is, sort of the greater effect it has on things like microbial biomass and diversity. And you do see significant reductions in things like, fungal diversity, bacterial diversity post-fire, especially when it's high intensity. So there could be something like that going on. With lower intensity fires, you tend to get these pulses of nutrient availability, actually. But again, that can kind of change, I think, as the fire gets more intense. Depending on what's around too, you can often get things like, and I know a lot of people are really concerned about this now with the fires, but you get things like hydrocarbon contamination and things like that. But in this case, I would I think in terms of like a burn pile, it could certainly be from a microbial perspective, but it's hard to say for sure. Yeah.
Thank you.
Yeah.
Okay. Next question was from Kessel Karen.
Yeah.
And I was trying to answer it, but the question was, did you say pair baiting, and is that literal? And I can, either Chelsea or I can answer that. And it is literal. We literally use pears as the bait. They're attractive to the oospores, right? Chelsea, help me out here.
The they're oomycetes. So yeah.
Oomycetes, thank you. They swim to the pear, and so the pear will show signs. There are certain signs that the pear will show, and so yeah, that's used as like the attractive bait.
Oh. I noticed it looked like there were pears in each bucket. So
yeah, so you soak the pears in them, and then you let them sort of air dry for a little while after that for a few days, and they'll develop symptoms. Essentially, if they're a Phytophthora there, they'll also develop symptoms but the symptoms look different, like for things like Pythium, like other pathogens as well. Pretty cool.
yeah.
All right, do we have any other questions? Feel free, if you'd like to unmute yourself, you're welcome to ask. Otherwise, just want to thank everybody for attending. And Chelsea, thank you so much for sharing all the work that you have. I'll give one more minute just in case I don't want to miss people's questions, but. All right, well, let's go ahead and give Chelsea a silent applause or a clap. Thank you so much. Really appreciate you sharing the work that you're doing, and it seems really important for our native plant communities and their success. So thanks for joining the Milo Baker chapter today.
Yeah, absolutely. Thank you. Thanks for being such a captive audience. And if you want to reach out and follow up about anything, I'm realizing now I didn't put my contact information, but you can find me at the Point Blue Conservation Science website. My contact information is on there. Or, reach out to Leah if you have hers and she can put you in contact.
All right, great. I forgot one announcement for our chapter, and that we have our general elections coming up for the next 2 years for our board, the Milo Baker board. So be sure to look out for that opportunity to vote for your president, vice president, secretary, and directors at large. And thank you.
And if anyone's interested in being on our board, please let Leah know. She's pulling together some names. So if you have any nominations or you'd like to serve as well, we'd love to have you.
Absolutely. We have a fun board, so come join us. All right, everybody, have a great rest of your evening, and we'll see you next time.
Okay, thank you.
Bye.
Bye.